Hi, I'm Dr. Arup Acharjee from Proteomics Lab, Department of Biosciences and Bioengineering, IIT Bombay. Today I shall talk about COVID-19 vaccines and how their vaccines are the ever-evolving final frontier against the war on COVID-19. There are no winners of war, only survivors. They only physically survive to warn us. Given the scale of pandemic, no quote holds true like this. We are going through a very tough phase of this pandemic. The second wave has engulfed the entire nation. And this is a war. We have been fighting this war ever since the pandemic evolved. And this will be a long drawn war. But every war has an adversary. Who is the adversary in this war? The adversary in this war is the coronavirus that we know as SARS-CoV-2. Now, what are coronaviruses? Are they extremely new? No, they're not new. Coronaviruses are known for pretty long. There are a large family of viruses, and they cause illnesses not only in humans, but also in animals such as bats, camels, and civets. We also have human coronaviruses which cause mild illnesses like common cold. And what are these viruses? Viruses like 229E, OC43, HKU1, and NL63. However, there are a group of coronaviruses that cause severe illness. Coronaviruses like SARS-CoV, MERS-CoV are known to mankind, and they are known because they have also caused epidemics. Previously, in the past 2 decades, we had two epidemics. One was the SARS-CoV epidemic, was the MERS-CoV epidemic. Today, we are facing another pandemic. This time, it is SARS-CoV-2. And it is a novel coronavirus because it has not been reported earlier in humans. So what are coronaviruses? So why is it called a coronavirus? So this family of viruses belongs to a family known as Coronaviridae of the order Nidoviralis. They're called corona simply because they're shaped like a crown-like spikes. So they have these spike proteins and they make it look like a crown. They're enveloped viruses and very small in size. The reason we need to use a very good quality mask to prevent uh, us from getting infections. And they contain RNA as their nucleic material, and their genome is usually between something between 26 to 32 kbs. SARS-CoV-2, where does this this virus originate? Nobody knows for sure. But what people believe, what we have few evidence pointing to that this virus probably originated in bats and via some intermediate animal host, which could be a domestic food animal or a wild animal or a domesticated wild animal, which nobody knows yet. The virus gained entry into humans. This phenomenon of a virus getting entry from an animal to a human is called zoonosis. So how does SARS-CoV-2 actually look like? So let us understand the virus a bit before we get into the vaccine part of it. So the adversary that we have today, the SARS-CoV-2, is primarily just like any other coronavirus. So it is an envelope virus. It has an envelope. It has an, if I can just use my pointer. So it has a membrane protein, a nucleocapsid protein that kinds of encapsulates the viral genome, and the spike protein. This spike protein is very important for for the virus to gain entry into our cells. And how does the genome look like? Well, a larger part of the genome is dedicated to the genes like RNA dependent RNA polymerase, helicase, proteases. So a larger part of the genome you would see is fairly called the non-structural protein genome part. They produce non-structural proteins, proteins which have no structural role to play in the virus, and they are basically parts of the genome that are very essential for the survival of the virus. Whereas a small portion, but a very significant portion of the genome is dedicated to structural proteins like spike, envelope, membrane, nucleocapsid. Now, these are structural proteins, so you really do not need uh, to be this genomic region does not really need to be highly stable, so to speak, because you see, if a particular gene that codes for your hemoglobin synthesis goes wrong in your body, you won't have any, you would really die. You might have a very severe disease like sickle cell anemia. But suppose you have 6 fingers, you won't really face much of a problem. Yeah, it would look odd, but again, at the same time, that mutation won't create any hurdles. Suppose you have a mutation that gives rise to a finger less. Again, you might have some problems, but those are not life threatening. Act 2 brute. So this is a scene from Julius Caesar. Brutus was a friend of Julius Caesar, and somehow he conspired to kill Julius Caesar. So why am I showing you this photograph? The reason is our body has a protein that somehow allowed this coronavirus to gain entry. So let us understand. So three proteins in our body, TMPRSS2, ACE2, and furin, are primarily responsible for why we get COVID. What happens is the viral spike protein comes and interacts with ACE2. ACE2 is angiotensin converting enzyme 2, which is very essential for our life. We need this protein, but at the same time, somehow it acts as the receptor for SARS-CoV-2. Once the spike protein comes and binds, the binding is happening in a region that we usually call the receptor binding domain. Once it binds, what happens is there are two endogenous proteases, furin and TMPRSS2, that comes and cleaves the S1 and S2 regions, subunits of the spike protein receptor binding domain. Once this cleavage is done, it allows for the penetration of the virus into the cell. Once the virus penetrates, it leads to the disease we know as COVID-19. And the fallout is very dangerous, to say the least. Millions of people have died after contracting COVID-19. If you look at the numbers are staggeringly high. Sometimes I really feel, how did so many people die despite the strong advances in science, the magnificent advances? We are sending satellites out in the space, and yet a small, minuscule virus is killing us in this course. Do we have a silver bullet to fight this war? Yes, we have. Vaccines. Vaccines are the silver bullet, that proverbial silver bullet that can help us win the war. So, what are the vaccines? What are these elixirs, so to speak? So, there are three main vaccines that have been produced in the world today. Although there are many, I will discuss primarily three, because, and the fourth vaccine that I would be discussing is Covaxin, that India is producing in large numbers. So in this vaccine, Pfizer, Moderna, and AstraZeneca, they have a common theme. So I was just telling you that the genome of coronavirus is around 30 kb. So it has all the information required for the virus to attack a cell, replicate within the cell, and wreak havoc inside the cell. Of these, the spike protein The key to enter into the cell, that spike protein region, is only 3,831 letters. So it codes for the proteins required for the virus to enter inside the cell. Now, this code has been used by scientists to actually stop the virus from gaining entry into the cell. How does it happen? Moderna and Pfizer are basically mRNA-based vaccines. They take the RNA molecule, the code is converted into RNA, and it is encapsulated in an oily membrane so that it can easily enter inside the cells. The Oxford vaccine or the Shadox vaccine or the Covishield vaccine, they're all the same thing, or the AstraZeneca vaccine. So these vaccines, this vaccine basically contains an adenovirus, a deactivated adenovirus that causes common cold in chimpanzees. And that acts as a mode of delivery of the code, which is now translated into DNA. Once it enters the cell, the Moderna, Pfizer, and the Oxford vaccine, what happens in the Moderna, Pfizer causes RNA, and it enters into the cell. The RNA is converted into protein, the spike protein, and Oxford vaccine, it enters as the DNA, transformed into the RNA, and again it produces viral protein. Once the cell, like our own cell, starts producing the viral proteins, these proteins are taken up by dendritic cells. Now, dendritic cells take it up and introduce it into our lymphatic system. Once it enters into the lymphatic system, a strong immune response is mounted because our body recognizes these proteins as something that is coming from the outside. They immediately pounce upon this protein and produce B lymphocytes and T lymphocytes. So B lymphocytes, they generate antibodies against the spike protein, whereas T lymphocytes will destroy any cell that is infected with the spike protein. What is the technology behind Covaxin? In Covaxin, simply the SARS-CoV-2 virus is allowed to propagate in a cell culture. Then there is an inactivating agent that is added on to the cell media, and all the viruses are inactivated. This inactivated virus is now injected along with few adjuvants inside our body, and it mounts an immune response. What is this typical immune response that we always talk of? Even in plasma therapy, people talk of neutralizing antibodies. So what are these neutralizing antibodies? Let me show you one small animation. So suppose this is the virus. Under normal circumstances, this virus will come and bind with the ACE2, which acts as the receptor. Once it binds, there is a cleavage, as I had just previously demonstrated, that will come into the cell and get infection. It will create an infection. But what if there are antibodies? The antibodies will come and bind with the spike protein. Once the spike protein is bound with antibodies, the spike protein will fail to interact with ACE2. If it fails to interact with ACE2, entry will be prevented. If entry is prevented, there is virus neutralization, and then there is protection. But is there any difference between the Moderna or the Covishield vaccine and the Covaxin? Yes, there is a fair amount of differences. So first difference, you might have noticed that these are spike protein based vaccines. So there is a strong neutralizing antibody response against the spike protein. There are also a T cell response, but again, against the spike protein. Whereas in case of inactivated virus, there is a neutralizing antibody response against the spike, but there is also a generation of a substantial amount of binding antibody against all viral proteins. Also, the T cell response is not only against spike protein, it is against all kinds of viral proteins. So be it ORF1ab, be it nucleoprotein, be it the helicase, be it any of the non-structural proteins, there is a strong T cells response. Now, does that mean it is game over for the virus? Many people would like to think so, but sadly, it ain't over till it's over. The second wave is a grim reminder that the virus is still amongst us, and many countries are actually having their fourth wave. So let us look into the evolution relationships of SARS-CoV-2 viruses during the ongoing pandemic. If you notice, we have almost 12 clades of the virus today. A virus that started off just as 19A has now evolved and you know become a virus that is now being called 20J. Now, this is a phylogeny of almost all the viruses that have been sequenced till that all the coronaviruses that has been sequenced from day one to the later part of the first part of April. And if you look at the distribution, if you would see no single country has, every country has a different distribution. The type of viral strains circulating among countries is pretty different. So virus has evolved independently in each country. So what started off with from China as the Wuhan strain of novel coronavirus or human coronavirus, now we have UK strain, now we have South African strain, we have US strain, we have the Indian strain. This is because the virus is continuously evolving. But does the viral genome evolve at a similar pace? So is it that the viral genome is evolving at very specific regions at very specific speed? No, the viral genome is fairly stable when it comes to the non-structural protein part. So this initial part of the genome, you would see there are regions where the it's fairly stable. This is a measure of the entropy or the number of mutations that you see. Now, if you see the entropy levels are pretty low in ORF1b, also very less in ORF1a. But if you go to the spike protein region, all hell breaks loose. Spike protein mutates like crazy. We will try to understand why it does that. And then again, nucleoprotein and like entire non-structural proteins are actually prone to mutation. As I told, if we have a problem in one of our very important organs, suppose I have a malfunction in my lungs, if I have a malfunction in my heart, my kidney, and my brain, I won't survive. I will die. But if the malfunction is that I have one finger less, I chopped off one finger due to an accident, or due to an accident, a part of my leg got chopped off. So these are like the structural part of my body. Then nothing, yeah, I would have difficulty surviving, but yes, I will survive. I won't die. So something similar happens in case of the viral genome. So if there are it's not that mutations do not happen in this region, or mutation only happens in the spike region. Mutations happen fairly across the board, but the mutations that survive, that pass on, that amplify are those mutations that happen in the structural protein region rather than the non-structural protein region. And how has the diversity of the viral genome changed over time? It has changed. The word that I would like to use is dramatic. If you see this representation of frequencies of clad over time, you see initially it was 19A, 19B, then it became 20A, 20B, 20C. Now we are in 20I, and this is 20J in the later part of April. So we are these 12 clades over a period of time. 19A and 19B have this Wuhan strain of 19A has almost gone out of circulation. So the virus we are fighting today is definitely not the same virus we were fighting a year ago. This is the same graph in another method. The way the representation methodology is different, but if you see the genome, how it has evolved over time, you would see 19A during 2020 Feb, but you don't see, you just hardly see 119A in 21 April. Most of it today is 20H, 20I, and 20J clans. So this is a reason for worry that we are with every passing year, we are fighting a new virus. The reason we need to vaccinate almost everybody so that we do not allow the virus to replicate. Each single round of replication allows for a mutation, and any bad mutation that will, I mean, bad for us, good for the virus, because it gives it a survival advantage. It can, attack more hosts, and it can, spread its wings, so to speak. So what are the drivers of viral evolution? So what scientists have understood till date? Why does this virus evolve so hard? The first primary reason for of viral evolution obviously is the number of replication cycles that happen over time. So more people getting infected means more chances of mutation. But that is something we cannot prevent, so to speak. But there are few events that have actually driven, or we have created natural selection pressures and selection pressures that has led to even more mutation, especially in the spike region. One is plasma therapy. Now, plasma therapy was touted, and even today many people believe that plasma therapy will save their lives. It's not true. A WHO study has nullified that there is any statistically significant advantage of giving plasma therapy. So these are, off-label use. So that all the plasma therapy you see going around is off-label, which means it does not have any scientific backing. So what harm does plasma therapy do? Now, this is a typical case study, and was published in Nature last month, in the month of April 2021. It is a case study where a patient, an old patient, was in the hospital for more than 100 days. So during the first 57 days, he did not receive any plasma therapy. And what scientists noted was the viral population, or the viral genome that the patient was having within his body stayed almost normal. There was not much of a difference. However, after convalescent plasma therapy, they observed a dynamic shift in the viral population. It's like something happened to the virus, suddenly the virus started changing. And why did the virus change? It started changing in the spike protein region. So there was a substitution, D796H in the S2 subunit, and the deletion in the S1 N-terminal domain of the spike protein. As passively transferred serum antibodies diminished, viruses with escape genotype were reduced in frequency before returning to a final unsuccessful course of convalescent plasma treatment, which basically means the passive immunization allowed for this mutation to grow. And also, what was not only did we have mutations, they also found the deletion mutation in the spike protein caused a twofold higher level of infectivity than wild type SARS-CoV-2. And also, there was a possibly compensation of reduced infectivity by the D796H mutation. So these mutations caused the virus to become twice as infective. So what this data, this paper, this case study tells us is that there is a strong selection pressure applied on the SARS-CoV-2 genome during convalescent plasma therapy, and that leads to emergence of viral evidence. And these, emergence of viral variants, and these variants are actually they have a reduced susceptibility to neutralizing antibody. Not only this, there is a fair chance of getting a mutation when the virus jumps host. Remember, we had discussed zoonosis, that there is a phenomenon called reverse zoonosis, where again the virus jumps from humans to animals and back to humans. I will come to that. So this is a wonderful paper which studies how the mutations have arisen from human to human transmission and human to animal passage. Now, if you would notice, there are animals like gorilla, dog, cats, lion, mink, ferret, mouse, and hamsters that have been infected with SARS-CoV-2. And when each time there is a passage, there is either a mutation in the receptor binding domain or the N-terminal domain of the spike protein. And not only this, I mean, there have been multiple research papers that have showed that coronaviruses have the susceptibility to affect ferrets, cats, dogs, and other domestic animals. I was talking about reverse zoonosis. What happened in the Netherlands during April and May 2020 is a grim reminder that can often happen anytime during the pandemic in any nation. Scientists noticed that there is an increased number of infections in mink farms. When they started genome sequencing and studying this, in depth, that why this infection is happening, they found that minks got infected with SARS-CoV-2, and later on, these infected minks were actually infecting humans, their human handlers. And where did the minks get the infection from? Obviously, from their human handlers. So the human handlers passed on the virus to the minks. The virus jumped onto the minks, infected the minks, and then the virus came back from the minks onto the human handlers. There was mass scale culling of minks, and somehow, as of today, this variant has gone extinct. We don't have this variant anymore. But does that mean that the phenomenon is extinct? No. Zoonosis and reverse zoonosis is a very likely possibility. So we need to be not only alert about the types of strains circulating in humans, we also need to study what are the strains that are circulating, in the cats and dogs and the feral animals that are going around in our society. What happens during this? There is a genetic term called bottleneck, genomic bottleneck. So think of it like this. A cat get infected with 5 or 10,000 viruses. Now, of those 10,000 viruses, there are 2 or 3 viruses which have a very strong affinity with the ACE2 of the cat. Now, which basically means these 2 or 3 viruses out of 10,000 viruses will be affecting the cats more. And the spike, that particular spike protein mutant will grow inside the cat compared to other 9,998 viruses which were also present. Just those 2 viruses will be actually infecting the cat. And now inside the cat's body, we have lots and lots and lots of the mutant virus that can jump back to the human host. And we really do not know. It's always, it takes some time to understand whether a particular strain is highly infective or less infective or more, does it kill more people or kill less people? There is another phenomenon that scientists have observed. Scientists have observed that newer variants like B1351 and the P1 or the Brazilian variant, they tend to infect mice without the need for human ACE2. Earlier variants like B1 variant and the B117 variant, they did not infect mice. The reason they could not infect mice is the spike protein was drastically different from the human, and the mice ACE2 did not allow for proper interaction. But this is not true for B1351 and P1 variants. What this tells us that these mutations are not only enhancing transmission and allowing for antibody evasion, but also expanding the host range. Now, this brings us to this question: do we also vaccinate dogs and cats against coronavirus? Yeah, theoretically, we should go for that. But practically, we are not being able to find vaccines for humans. So this virus will keep on spreading amongst animals. And again, there will be a phenomenon of zoonosis when we would have another variant of SARS-CoV-2 that can evade the antibody generated during vaccination or by vaccination or by natural infection. So these are dangerous times we are living in. So what are these phenomena called? So these phenomena are called antigenic drift and antigenic shift. So what is an antigenic drift? It is basically a very small minor antigenic change, and it leads to a new viral strain. So whatever today we are seeing is basically antigenic drift. The UK strain, the South African strain, the Indian strain. So what happens is a new viral strain may infect the host of the same species. Now, in case of antigenic shift, and it is very, it gives rise to epidemics. Usually, what people have, earlier studies have indicated that antigenic drift can cause epidemics between pandemics. But today we are going through a pandemic, and that leads to antigenic shift. Now, what is an antigenic shift? It's a sudden shift in the antigenicity of the virus. So basically, the entire antigenic profile of the viral proteome changes. So two different strains of the viruses combine to form a new subtype A, B, C, whatever. And this results in a major antigenic change. And it is rare, it does not happen all the time. But when it happens, it is very difficult to treat. And it might give rise to newer pandemics. So we have to be very, very alert regarding SARS-CoV-2, given that it is rapidly jumping from one host to other, given that it is infecting so many people. We are on the verge, we are sitting on the edge of a cliff of antigenic shift, which basically means we have a new enemy at the gates, and we better be prepared for that. What happens when there is antigenic shift and antigenic drift? In Manaus, Brazil, it was observed that almost 50% of the population had antibodies against SARS-CoV-2. This was a seroprevalence study done, just like India is doing, just like many other countries are doing. But despite the high seroprevalence, suddenly in late December 2020 and January 2021, there was a sudden spike in the number of deaths and the number of COVID-19 hospitalizations. When scientists started studying that what might have gone wrong, they understood that this is a new lineage of the SARS-CoV-2 virus, and that is known as the P1 lineage, or the Brazil variant, Brazil mutant. So this is what new variants does. Despite having antibodies against the previous COVID, previous SARS-CoV-2, they failed to protect people from hospitalization and death. And if you see over the past 6 months, maybe late uh, early November to today's April, there has been a surge in the number of emerging lineage strains. So these emerging lineages that includes P1, P3. So P3 is another new lineage. It also has the triple mutant from India, this double mutant from India, B1617. So all these emerging lineages have actually come in the later part of 2020 and the earlier part of 2021. No wonder we are facing a second wave. Despite the high seroprevalence, we are facing these newer strains of the virus. And maybe even if somebody had an asymptomatic infection during the first wave of COVID, that is not saving him or her from this second wave, because this is a new enemy. So what are those variants of concern, so to speak? So there are multiple variants. So there is the UK variant that has multiple mutations on the spike protein region. It is thought to have 40% more increased transmissibility. And currently, what new studies have shown that there is no reason to worry when it comes to the vaccine efficacy. Then we have the B1351 South African variant. Again, they have 21 mutations in total, but there are three mutations on the spike protein. And in vitro studies have suggested there is a potential for immune escape. Again, there is the P1 variant or the Brazilian strain. One common factor is multiple. There are three mutations on the spike protein, and there is a high chance. I mean, there are anecdotes and now reports are coming in that there is a lot of reinfection, which basically means there is immune evasion going on. There are a few strains that are reason for concern in the US. And today in India, we have the mutation, which is famously called the double mutant. The double mutant has two very unique mutations at the spike protein region. Overall, the spike has three mutations, and the vaccine efficacy is also a cause for concern. So you see, every country, every region is starting to have their own strain, which might cause a reason for concern because they are distinctly different in their pattern in which they spread. They're also concerned whether the existing vaccines would be working. So scientists did a very wonderful experiment where they wanted to understand how coronavirus immunity works, because this is very pertinent if you want to learn. We cannot wait 10 years, 15 years to understand how COVID-19 immunity would be behaving. So what scientists did was take lessons from previous coronaviruses. But what we understand today is humans are repeatedly reinfected with common cold coronaviruses. And studies have suggested that a typical person is infected with a specific type of coronavirus. In this case, the study was on 229E every 2 to 3 years. Scientists also did a study to understand if a previous coronavirus infection can prevent a future or future variant of the same virus. What they did was they collected human sera of the 1980s and the 1990s and checked for neutralizing titers against contemporary 229E coronavirus. What they found was coronavirus of the 1980s was easily neutralized with the serum of 1980s. But if you want to neutralize a virus that was in circulation in the 1990s with the serum of 1980s, it did not work. So there was a difference between 8 to 17 years that they found on an average, that there was so much antigenic evolution of the viral spike that future viruses could not be neutralized. So as I said, viruses of 1990s failed to get neutralized by the sera of 1980s. So what the scientists extrapolated from this study is that it is advisable to periodically update the SARS-CoV-2 vaccines. Since we are talking about wars and fights, one is intrigued that how do we win this war? Is it like this perpetual thing that is going on? People will keep on dying? No, we can win this war. How do we win this war? Surveillance, community viral surveillance. So this is a paper in Science that tells how people in England were recruited into a nationwide self-administered SARS-CoV-2 test. So people took self-administered the swab and sent it to the lab. And then they started, what they observed was they could actually predict when, when there is going to be a community flare up. So we need to adopt technologies, strategies like this, where we take random samplings from communities across the country and do, random PCR studies and try to check whether there is a, increase in infectivity, sudden increase in infectivity. Because if there is a sudden increase in infectivity, which it basically means that you are having a community flare up, and then thereafter, you can use more advanced tools like genomics and proteomics. So what with genomics? The weapons of the future are the next gen sequencing. So honestly, we must thank next generation sequencing for saving us. Next generation sequencing, overall DNA sequencing technologies have undergone a sea change over the past 10 years. It has become cheaper and it has become faster. Now, the entire vaccine market must thank the next gen sequencer in Wuhan, which sequenced the viral genome in almost less than 15 days after it was isolated. And that is how we came to know that this is a new virus, that this is a novel coronavirus. So applying next generation sequence strategies, we need to sequence more and more viral genomes. And then we have systems like the GIS-AID and NextTrain. What do they do? GIS-AID is an open source, global influenza surveillance and response system. They collect genome data from across the world. So if I am sitting in IIT Bombay, I do a sequencing of a COVID genome. I will submit my genome sequence data to GIS-AID. Once the data is there on GIS-AID, GIS-AID will study that how my genome compares with the existing genome. It will tell me about the evolution. So much of the figures that we saw in the earlier part of the talk was taken directly with the help of GIS-AID and another tool called NextTrain. Now, NextTrain is another software tool that has been developed to check, or rather to study the evolution of the virus. And not only about SARS-CoV-2, NextTrain also explores pathogens like seasonal influenza, West Nile virus, Ebola, measles, dengue, you name it, they have it. But not only genomics, we need to apply techniques such as, proteomics techniques such as protein microarray. What is a protein microarray? I'm giving you the plan. I'm just showing you how this experiment can work through. So once you're infected with the coronavirus, you will produce antibodies against the coronavirus. Now, these antibodies recognize specific portions of the coronavirus proteome. That those specific regions are called epitopes. Epitopes are regions which bind with the antibody. So the antibody can recognize a specific part of the protein that is known as the epitope. Now, what we can do is study the epitope pattern. We can do a epitope fingerprinting. We take serum from a patient who has been infected with a new strain of the virus, and we try to create a map of what are the epitopes of the antibodies this person has generated. Once we get that map, we can compare it with the map of a person who got infected with the earlier strain. So we have a map, antigenic map or epitopic map of the Wuhan strain, and now we take the epitopic map of the Indian double mutant strain, and we try to match, and we try to see if there is any shift in the epitope. How does it help us? It might help us in identifying strain specific epitopes. Once we identify this is epitope is very strain specific, we can use that information to design rapid antibody test, which we can administer to patient to check for that strain. Now we can design an antibody test for the UK strain. We can design an antibody test for the Indian strain. Also, it will also give us a fair amount of idea whether the antigenic profile of the virus has changed. Does it, will it respond to the vaccination that has been administered to the patient, or the neutralizing anti with the neutralizing antibodies work in this strain? All these information we can gain from a protein microarray. But do we need to worry? Perhaps today we do not need to worry much, because there are multiple reports which show that the existing vaccines like Covaxin and Covishield from India, the Pfizer vaccine, the Moderna vaccine, all of them seem to work well to prevent us from getting infected with the variants. But at the same time, there is a warning. There are studies which have shown that people are getting reinfected with COVID. And these reinfections are happening from a phylogenetically distinct strain. So this is paper reports that there is symptomatic severe acute respiratory syndrome, reinfection by a phylogenetically distinct strain. Then there is recurrent COVID-19 inducing evidence of reinfection and enhanced severity in 30 Brazilian healthcare workers. So all these reports tell us that we have to be very alert when it comes to fighting this war against COVID. With this, I would like to end my talk. I would like to acknowledge Professor Sanjeeva Srivastava from IIT Bombay, my lab members from Proteomics Lab IIT Bombay, Dr. Radhika Vaishnav, who has been helpful and in critically reviewing these slides, and Professor Royana Singh from Institute of Medical Sciences, Banaras Hindu University. So this is some food for thought before we end this talk. Thank you for your patience listening.